Hello, everyone, and welcome to today's live broadcast, an ongoing evaluation of our transient expression systems. Can we predict the appropriate expression system for any drug modality? Presented by Richard Altman, a scientist in the Protein Technologies Mammalian Expression Group at Amgen San Francisco. We are excited to bring you this educational web seminar presented by LabRoots. This event is sponsored by Thermo Fisher Scientific, and this live broadcast is brought to you by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your questions into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. And if you have any trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Please join me in welcoming Dr. Richard Altman. I will now turn the presentation over to him.
Thanks, Kristen, for that kind introduction. First, I'd like to start with the important things, and that's thanking the organizers for the invitation to participate in this excellent online conference, both for the invitation to speak and then also especially to Rick Bassey for the invitation and then also for the organization of the conference. And also I'd like to thank Henry Chu and John Zamuda for their assistance in organizing this as well. So I've shortened the title a little bit from what Kristen had read to Can we predict the appropriate expression system for any drug modality? And the short answer, if you want to skip the rest of the presentation, is we're not there yet. But what I'm hoping that we can do as we share the next several minutes together is sort of see what the importance of this question is and how we're going about answering it with some experimental data so far and what we're planning or what we're looking forward to. And then also tying in some ideas of not only answering this question, but about how we increase the efficiency of our protein expression process. So I think, as you see on the introduction here, with an ever-expanding array of protein formats, really one of the next larger challenges that we see is being able to strategically match a construct with an expression system. And I think this will become a little bit more apparent as we go through some of the subsequent slides. In order to really take a look at this experimentally, what we've done is to utilize a diverse set of proteins, comparing material that we've transiently produced in a number of systems, including the HEK2936E, the ExpiCHO, and the ExP293 systems, both for expression level and product quality. And I think I want to stop there and make it a special point about the product quality. I think, and again, as we'll see in future slides, the titer has been the focus for many of us in the expression field for a number of years. But I think as titers have increased, the idea of product quality has taken a more formidable sort of position at the table in terms of important attributes as we go about fulfilling requests and generating proteins. And what does this allow us to do? And hopefully what it does is we can prioritize expression systems. We can maybe potentially limit the number of expression systems we have. And what we can do there is reduce parallel effects and increase our throughput efficiency. And with that increased efficiency, there are some subsequent efforts that I'd like to talk about at the end of the talk that increase our efficiency of the whole process. And again, I think that's an important topic as well when you look at increasing efficiencies, is not just the expression portion of it, but if you look at the whole process really from cloning through analytics, how do we reduce that total cycle time in the whole process? So I think one of the fundamental questions I always suggest is that we take a look at what do we want to do, what do we want to establish. And one of the things is establishing or optimizing these systems to meet the demands of our protein production. And one of the terms I like to use often is strategic. This strategic selection of expression system. And I'll define this a little bit more in a subsequent slide. But really, the main goal is here is how do we build an appropriate expression toolbox to support drug discovery efforts? And there's no right or wrong answer here. I think that it's going to be, certainly everything is going to be specific to your particular expression lab or expression facility. But I think it's an overall idea that we need to continue to focus on. And I think the constraint or sort of the border that we have or would like to live within about this is that when we talk about this, when we talk about looking at systems or optimizing systems, that we don't want to lose sight of this term of ease of use. And I think it could come down to a risk-reward or how much effort you're putting into something for the reward outcome. And I think for any of these things, as scientists, we always think we can tweak another 1% or 2% out. And in some cases, that's very important, and you want to do that. But I would argue in a lot of cases this isn't necessary. And so when we go through these processes and try to carve out niches of time from our day-to-day work to methods optimization or looking at different methods, that we keep that as sort of a guiding light for us. So in today's discussion with everyone, I'd like to look at this evaluation optimization of transient expression systems. And we're going to talk about strategic selection of expression systems and cell lines. So we're going to look at a little bit, give a quick review of the expression systems that we have here in our facility. What are their advantages, what are their sort of characteristics? What advantages do they bring to us right now? And how are we doing in terms of getting to the point of assigning them specific formats for specific expression systems? Then I want to jump gears and talk about scalability, and maybe not in the sense that the traditional sense, but in a couple of different ways, and I think that will become apparent in a few minutes. And then also talk about optimization. And both the scalability and optimization are going to come back into this whole efficiency topic in terms of how we choose systems and how we do it most with the quickest turnaround time. And then we want to step back and look at a couple of addressable process challenges that I think are interesting for facilities in general about how we go about some processes that are fairly time-consuming, but we hope to have some leads on how we make these sort of time-sink projects or time-sink portions of the process more efficient for us to allow us to do other things. So let's go to that first bullet point and talk about strategic selection of expression systems or cell lines. So we might ask the question, well, why is this important, or why should we do that? And I think this slide is a good start or a good graphic representation of that. I think when we think back into years, 15 years or so ago, when we look at what we were expressing, and predominantly I believe we were expressing a lot of just traditional monoclonal antibodies, which you can see there on the panel on the left. This panel on the right is from a recent review article in 2015 from Spice et al., which is showing a number of formats of bispecific antibodies. And as the protein engineering has advanced, so has the number of formats. And each of these, I think in their own way, can represent challenges for us in terms of expression levels and product quality, and in terms of maybe one size or one expression system doesn't fit all of these. The following couple slides are from a review article from Yager et al., which they took a look at all of the cell lines that have been used for transient protein expression. And here on the first page, you can see a number on the left-hand side, the left panel, of 293 based cell lines, and then starting in midway down on the right-hand side, a number of CHOs. And this continues onto the next page of additional CHO lines, and then a couple of newcomers, or not necessarily newcomers, but others that are added in there, including the CAP T cells, the immunocyte-derived cells, and then also the Vera cells, the African green monkey kidney cells. But I think when we really think about it, when we look at all of these things, it really sort of boils down to, at this point, some commonly used lines for transient protein production, and that really centers on HEK293 cells on the left and CHO cells on the right. And I borrowed this slide from Henry Chu, who has presented this before, but it is a great summary slide of the advantages of both. You know, for the 293s, obviously for years and years, these have been the cell lines of choice. They're highly transfectable. They've given excellent titers. They have been the cell line of choice. But many of us in the protein expression world, since most biotherapeutics are produced in CHO-derived cell lines, have always hoped for a CHO line that we could use in a transient paradigm that would allow us to create material that's more representative of what that final product is going to look like from a production line. But it's been difficult with the transfectability of CHO cells. But with advances in both methods of transfection in terms of electroporation, and then also from the launch of the ExpiCHO system, we now have some choices that we can generate material with post-translational modifications that are more in line with what we're looking for in a much more transfectable cell line. What I would say is one of the challenges that we have here with our choice is, do we always have to go into CHO cells? And I would say the answer there is no. I think there's still a lot of applications that we can use 293 cells. And I think, as I said, I'll point out some advantages of that a little bit later in the talk. But now we have that additional tool of the CHOs when we need to use it in a transient setting. And this is just to remind myself, and I think and everyone else, this just represents, from a recent paper, sort of the pathways and barriers to gene delivery. And really, on a sort of very high-level point of view, the difficulty and the complexity of the whole process, and what we need to do to sort of, at every step, whatever we can control and optimize to help that, to help the whole efficiency of the process is important. From the choice of transfection, whether we're going the lipid route or electroporation, to the health of the cells, to conditions like that. It's not meant to be a depressing slide in terms of the complexity or variabilities, but I think just a reminder of how fascinating and complex this whole process really is. So when we talk about here, to get into the evaluation of the expression systems, when we talk about the cell line strategic selection, what I mean by strategic, as I've already spoken to, is can we predict which expression system will give the optimal titer and protein quality for any protein format? And you can tell from all of those formats that we have, I'm not sure that we'll ever be able to 100% do this. In fact, I'm pretty convinced that we won't be able to. But I think what we will be able to do is to at least give us some educated ideas of what systems to go into. And you see at the bottom of this slide some of the transient expression systems that we routinely use here in San Francisco. We utilize the ExpiCHO system, the ExpiCHO system, and also an alternative protocol to that, the high-density ExpiCHO. And then we also use the 293-6E expression system, which historically has been valuable to us over the last number of years. Traditionally, we've used a freestyle F17 expression media with that system, but recently we have done some experiments in terms of equilibrating this with the balanced CD system from Irvine Scientific, and we're finding some encouraging results with that at this point. And they all have, if you look at a comparison of these systems in a sort of tabular form as in this slide, you can see here that when you look at, I've had 4 categories here of what I think are important considerations for these systems. There is a variety of the amount of plasma that's used for transfection in any of these systems. And one of the things you'll see an asterisk there is when we're talking about optimizing gene dosing in this expression system, in a particular expression system. This is a topic that we're going to speak about later in the slide, but as we do larger and larger transient expression runs, the whole idea of how much minimizing the amount of DNA we can use is a very important thing, both from a cost and from a process point of view. But you can see here we're running anywhere from about 0.2 micrograms per mil up to a microgram per mil. But we're especially interested in both the 6E system and the ExpiCHO system about doing some additional gene dosing experiments. And like I said, we'll talk about that a little bit later in the presentation. The densities vary in terms of the density at transfection. And they run anywhere from 1.5 to 2 million per mil with the 6E system, all the way to 10 million per mil in the ExpiCHO high density. And I think one of the important things for us is the harvest time. And you can see here the range that we're experiencing. With the 6E system, we're looking at about a week, 7 days of expression time. In the ExpiCHO system, the multiple systems in the ExpiCHO is running anywhere from 7 to 14 days, depending on the protocol that you run. But one of the advantages, I think, with the ExpiCHO system that we've seen time and time again is the ability to really harvest early at day 4 with a minimal level of loss of titer with that. So we can transfect on a Monday and harvest on a Friday and have a very quick turnaround time for a number of applications. And this sort of drove our interest and experimentally work into looking and validating more the high-density ExpiCHO system in terms of getting it down to a day 4 harvest. And we've had pretty good success with that. I'm not going to share any or show any data about that today, but I think as we look at in a broader picture how transients are stacking up against stable pool processes these days, I think it's an incredibly important consideration in terms of the days to harvest. Can we keep transients transient? And so I think pushing those back is certainly a goal of ours to the quickest sort of time to harvest. And then we talk about advantages, and the 6E systems have theoretically extended expression times due to the generation of the EBNA1 protein binding to the OREP in the plasmids. As we've mentioned in the 293s, that time to harvest is really great, both from the expression point of view and then also from the whole process point of view. And then also, as we've talked about in the ExpiCHO, the post-translational modifications are much more in line with what we're seeing for products that are made in CHO lines, therapeutics that are made in CHO lines. So we have all these systems in place, and now, so how do we go about sort of figuring out what each of them, what their strengths are, and how we can use them best? And I think that one of the things that we have found, I'll give you a quick thumbnail on these, is that with the ExpiCHOs, we see excellent titer and excellent product quality, for the most part, in the generation of regular mAbs. And as we'll circle back to later on, this is for us right at the moment, this is their strength. But the next set of the next set of slides are going to show the difficulty, I think, in trying to do this, because there's always exceptions to these sort of rules. And this is one of these cases where we had a traditional mAb. And if you look here on the slide, the panel on the left is a non-reducing portion of the gel. On that side, and then on the right is the reducing portion. And what we've done here is we've taken the same mAb, the same construct, and done a small-scale expression, 30 mil expression, in 293 6Es, in the XB293s, and in the XpiCHOs. These are different days of duration. The 6Es and the XpiCHOs are 7 days. The XB293 are 4 days. And when we look at this, when we characterize the material we're expressing, we look at octet when possible and then run on SDS page. And what you can see here is that if you look at the chart on the right, that when we looked at that the expression level certainly was enriched in the XB293 and XpiCHO over the 6Es. We were in the range of 400 to 500 in the XB systems and about 150 in the 6Es. But then the second part of that, talking about product quality, comes into play. And so when we looked at this on a gel, you can see here up in the 150 range, where now in the 6E and the XB293, we're seeing in this area at around 150 where we would expect this, the majority of the product being migrating. With the CHO, we're seeing certainly some of that material, but also we're seeing a level, if you look down the lane, of additional products being generated. And so this is one of those examples of, most of the time it is, but not true for the XpiCHOs. The XB293 here would be the system of choice, and that's what we did. And in the subsequent slide is showing a Western blot, showing here again that this is against anti-human IgG heavy and light, where again the product quality is clearly much better in the XB293 and to some extent in the 6E, except we have better titer in the XB293 than what we're seeing here in the XpiCHO. And this next slide just sort of reinforces the idea of the increased titers that we see with the XpiCHO. And if you look, what we've done here in that left side of the panel is there are the conditioned media titers and then the purification yield on the left side for 4 mAbs that were expressed in the 6E system, and then on the right side in the XpiCHO system. And again, as you can tell here, that the titers have been much more enhanced in the system, in the XpiCHO system, than in our previous system. And when we have done a couple of these in parallel in the in the high density system, and what we find is that the titers are very similar as what we're seeing in the 7-day standard protocol. So it's one of those switching slides here, or trying. Bear with me a minute. Yeah, actually, if we can get some assistance in changing that, that would be great. I'll continue to talk through this. I apologize for the problem with going to the next set of slides. What I was going to say is what I'd like to show on the next set of slides here is that what we've done is to take another set of molecules, and in this time, not just, thank you for that, not just MABs, but in this case also fusions. So if you look on the left side of the gel, we have non-reducing conditions, and on the right side we have reducing conditions. And we have 3 different fusion, monoclonal antibody fusion proteins, and then in the first 3 lanes here, in lanes 1, 2, and 3, and then a traditional MAB, and these are all in the ExpiCHO system. And you can see here, again, that they have all performed very well. We have differing titers. You can see in the ExpiCHO system that the first one is a little bit less titer of 150, but then we see excellent titers in the 400s, and then the MAB in the 500s, in the 700s. And you can see at the bottom with that MAB that we're seeing really good yields in this, that we're getting about 350, 360 mg per liter out of that. And from the purification point of view, we're seeing excellent in terms of excellent homogeneity in terms of the SDS page and the mass spec data. So just sort of reinforcing where a strength of the ExpiCHO system is. And then this represents, again, a fusion that we ran in ExpiCHO, and it's giving us a little bit more of a challenge. And what you can see, this is through the purification on the left there. The lane 1 is the load through, then flow through, and then off of the first column in lane 3, and then the elutions in 4 and 5. And if you look under reducing conditions here in 6 and 7 off the purified material, we're seeing a band at around 50 kD that really should not be there, right about there. And the mass spec is basically indicating to us that we're getting some clipping issues. And as you'll be able to tell as we're moving forward, this is one of the challenges that we're having with the system, balancing off the really advantageous part of the monoclonal production and post-translational modifications. Now this slide represents a protein that we use for assays, so it's a reagent protein production. But this was interesting in the sense of taking a step back. This was expressed in the HEK 293-6E line a long time ago, so there was some historical data with this. What we did, and I don't have the time to show you the data in here, is that we ran a comparison of the 6Es, the XB293 and the ExpiCHO. And with this particular protein, what we found in the past it worked with was with the 6E system and harvesting at day 3 because there was some susceptibility to degradation, and we saw that with the other cell lines as well. But this gave us the opportunity to look at this additional or this new media from Irvine Scientific in terms of the balanced CD. And one of the, from a presentation at Pep Talk recently, one of the advantages of this is that it's should give higher titers with the same cell. And so we wanted certainly to investigate that. And what we did was sort of a combination of looking at this system and doing a gene dosing experiment. And what I mean by the gene dosing, and like I said, we'll talk about this a little bit more in detail later, is to not only just reduce the amount of DNA, but to reduce your DNA of interest and keep the amount constant that's being transfected, but keep it constant with an amount of empty vector. And so if you look here at the slide, on the left, in lane 2, you see just an IG standard. And in lane 3, what you see here is a purified standard of the protein of interest, and that's right in there. And then we look through here, and what we're doing is we're doing both 0.2 micrograms per mil and 1 microgram per mil in triplicates here, where we're looking at mock, and then 0.2 micrograms per mil, 1 microgram per mil. And this is the control amount that we saw in the F17 media. And as you look across, what we see is that by the time we get to day 7, that the winner here for us really is the less amount of DNA at 0.2 micrograms per mil that gives us much more robust expression of our protein of interest, certainly as compared to the material. If we take it out in day 7 that we experienced before, we're seeing a lot of disappearing of that material. So if we run a Western on that, just to sort of reiterate that, you can see here this is an antibody against that protein of interest that we're seeing really excellent amount of immunoreactivity here with that at the day 7 at the 0.2 microgram per mil. So it gives you an idea of the amount of iterations that we're having or can use or bring to bear on these different systems or with these different systems and different media, and how it's so much an ongoing sort of process. So this is, as I've said, there are many more examples we can talk about of these side-by-sides, and what we're doing here is not only with new experiments taking a look at comparisons, but also we're historically going back and looking at material that we've generated in these different systems and really trying to, with a high N value, come back and say, how are we going to prioritize our systems? And this just shows sort of where we are at this point with what we look at or how we look at our systems in-house. So I think from the system that we use the most is really the XB293 system. And its huge advantages are really the time to harvest, as we've spoken about. And what we've experienced is that it works with a variety of formats. And as I mentioned before, I think for most of the applications that we use it for, it's a perfectly acceptable cell line to use to meet the needs of those applications. Now, what are we doing in terms of optimizing that? Well, we talked about gene dosing. Generally, in the past, I think the recommended amount is a microgram per mil. I found we can go down to a 0.5 microgram per mil, and that's just a dilution of the DNA. There's no gene dosing replacing, keeping the amount of DNA constant and replacing with empty vector, but that's something we're doing right now. The 6E system, I think, because of the advantage it seems to be giving with the additional or different media, and its time to harvest is giving us is probably our next go-to. And I think a big thing there, as I've mentioned, is that it's historical use of the system. We've expressed a lot of proteins, and I'm sure many of you get the same sort of thing that you get refills or you get requests again and again for different proteins. And here we have a source. We have a source of an expression system that we can go back, and we know we've made this material, and it's been of good product quality. The ExpiCHO system is something that we are using. We feel well suited to traditional mAb formats, and it has the advantage of the post-translational modifications. We're working diligently to really characterize other formats and to see if between different cell line conditions and maybe different engineering of the construct, if we can't make that the umbrella of the ExpiCHO system even wider for our use. But I think at the position we're at right now, this is where we feel most comfortable with the use of the system. And I think when you put the three of these together, what it creates for us is a fairly good toolbox that we can address the needs of most of our requests, But as I said, there's still work to be done, and these orders might change as time goes on. I think an interesting point, to think about is how many systems do you need? It would be great to go down to one system and have it fit everything. I just don't think in the near future that this is a sort of viable sort of place, but that might change too in the coming years. But at this point, this works well for us. So I want to talk about scalability, and I think most of what I want to talk about is small-scale screening to support this expression selection. So, when we talk about how do we go about running these side by sides, and it's fine, we can run these in 125 mil flasks and do 30 mil expression studies. But one of the things that we've recently set up that I think is incredibly helpful for us is to do these at very small scale in a relatively higher throughput in a 24 deep well plate. And whether we're using a TCAN here and doing it in very high throughput sort of mode, or we're just doing it manually depending on the number of constructs, what we can do is transfect in these different expression systems and then harvest these volumes and then hand it off, take a look quickly at the expression analysis, whether octet or SDS page or even CalPER for higher throughput, and then hand this off to our colleagues in expression who have a high throughput affinity purification, either with protein A or nickel NTA or whatever. And then that expression time is about an hour and a half for them to do at this scale. And then we have a follow-up analytics where we can now get not only what's the expression level, but what's the quality of the protein. Are we seeing aggregation or whatever? And we've used this effectively for a few times in terms of then being able to, within a week or so, or less even sometimes, come back and say, really we should choose, expression system A for this particular format. It's also very viable in the sense of when you have something novel that we don't know titers, but we're pretty sure of expression system, it too gives us the ability to scale appropriately so that we're not just doing an educated guess, but we actually then, with just an extra week or so built in, then we can scale up appropriately so we're not overshooting or undershooting and creating additional work for ourselves. And then I think one of the challenges that dovetails into that is just having the ability to do the scales that are necessary. And you see here there's a whole variety, and we experience all of that from the deep well plates right to the wave bioreactors. And so having those in place are critical, but that small end really, I think, is going to speed up the efficiency of our process and allow us to make really good decisions moving forward. So we talked a little bit about optimization. And we've mentioned this idea of gene dosing. There's a lot of variables here that should be considered to improve expression levels and product quality. And we've discussed choice of cell line. We'll talk about gene dosing here a little bit more in a couple slides. But there's also many others, and I've just mentioned temperature, duration of culture. DNA quality, I think, is a topic that people are aware of, but probably we should pay more attention to moving forward. And then also with the production of antibodies, stoichiometry of the heavy chain, light chain ratios that we are transfecting with. But let's talk just a little bit more in depth about the gene dosing. And really, it's a useful strategy, not only for obviously saving DNA, but for generating difficult to express molecules. And we're basically what we're doing here is decreasing the transcript to match the secretory capacity of the cell. Mostly with the use of the empty vector, with all of the elements, the promoter elements and so on that are there in the empty vector, they're acting as a transcriptional factor sink and to sort of modulate the amount of transcript. So hopefully we can create a higher quality product that is less prone to aggregation. So again, varying the amount of coding DNA being transfected. And then again, the sort of a collateral benefit is it's a means to decrease plasmid needs. And this is an excellent example from a paper that was published a couple of years ago by my colleague Bram Estes and his colleagues down at our Thousand Oaks facility, where if you look at this, that gene dosing can improve the product quality, but if you look at the form of carrier DNA, it can make a difference. And a lot of times when we think about this, we think about that a lot of times we would use the salmon sperm or shared salmon sperm DNA, and as you look across, this is a fusion protein, and you look at the gene dosing percentages from 100 all the way over to 2. And with the shared salmon sperm DNA, you do see some improvement of this from all these multiple species here in the aggregation to something that's more amenable here, that's running about 95 kilodaltons. The same thing with this blue script vector in the middle panel. Again, you're seeing some improvement in terms of the aggregation properties, but in the panel on the left, where they used the specific vector that was done in the expression system, the PTT5 in this case, you see by the time you get down to 10% and 5%, that now we're generating a pretty good, much nicer product of higher quality. And this is something, like I said, that we're doing on an ongoing basis for both of those reasons, both for constructs that are generating material that are prone to aggregation or clipping or whatever, and then also as a way to reduce our DNA needs in the future. And in the last couple minutes, I just want to talk about the efficiency of the process and talk about a couple of things that we're doing to sort of address that. And I think this slide is a great reminder of this, is that if you think about it, it's not just about titer anymore. This is a figure taken from a recent paper where, back in the 90s, the thought of creating, or early 90s, the thought of creating a gram of material transiently was met with skepticism, to say the least. But now, by the mid-2000s, you can see that the ability to generate material at 5 to 10 grams per liter and cell densities of very high is not unusual anymore. So it really isn't just about titer anymore. It's about, like we said, the quality and then speeding the whole process. And how are we looking at that? Well, there's a couple of ways here, and I'm going to go to the second one first here, in the sense of the process efficiency between cloning, expression, and purification groups. I think this is something not to be overlooked. We won't speak about this in this presentation, but when you think about it, I think there's a lot of white space in between those times where constructs are generated and the material is purified, and how we go about, and communication is a big key here, how we go about getting these passing, doing handoffs and so on is really an important consideration. But I'd like to just talk briefly about some emerging technologies and really focus on the bioreactor harvest process, which, if we look at the next slide, traditionally, and I borrowed this from a presentation of my colleague Ray Lowe, if you look at it, when we've run stuff, especially this is true at higher scale, that we have the bioreactor, and then we're faced at harvest to do centrifugation and then subsequent filtration, and then column purification. And this whole centrifugation step, as many of you are aware, especially if you're dealing with 10 liters or 20 liters or more, is really a time sink, and it really ties someone up for a number of hours to get this through both the centrifugation and the filtration. Well, is there a way we can really sort of start to get around this? And the answer is yes, although there is not a rigid process in place at this point. This just shows here one of the ideas that we're pursuing, and that's of using, especially for large scales, these GE dual filter cassettes, where we're basically just hooking this up to a wave bag here, and then pumping through the filters both a glass fiber 5 micron and then a 0.2 micron sterilization filter, PES filter, and then into a sterile bag. But basically, once we make the connections with these, we can then walk away and people can be engaged in other activities in the lab while this goes ahead. But of course, this is not without some optimization and getting it, getting the right parameters in place. The first few times we ran this, we really ran into some issues about, I think, the speed at which we were pumping through the material, and we were really damaging the cells. And so we took a step back, did some optimizations on flow rates, and then did a study where we took 20 liters, took our pretty much our optimized, what we thought at the time optimized conditions, and then instead of harvesting into a bag, we took 2 liters aliquot, so to speak, and then looked at this both by octet and by SDS page. And what we found is that you can see here from lanes 4 on, these are just subsequent fractions, is that pretty much the product looks the same, and the titers, until we get right to the end, as measured by octet and as visualized here by SDS page, are pretty consistent. So I think we've found ourselves some parameters where we can harvest this reliably without much loss of material. And this is an ongoing process for us at this point. And a second sort of exciting thing that we're also investigating now is you see this traditional column process here on the left that we saw earlier. But then one of the things that we're working on in collaboration with our colleagues down in Thousand Oaks is this mag bead process, where we're taking magnetic beads that are coated either with protein A or some other affinity mechanism, and then putting these in the bag the night before harvest, letting them bind, and then just using magnetic rods, harvesting them out of the bag, rinsing and eluting. And the whole time here, is you can see in this panel, is we've shrunk the time down to the harvest being about an hour and a half or so. And not only just the harvest time, but what we've done effectively is a first step purification and a concentration step. And up here you can see the comparison between the mag beads and the MabSure Select traditional method. You can see similar sort of recoveries and yields and product quality in this system. Again, this is a sort of a work in progress, but again, it would allow us to sort of free up some time from this fairly mundane process to do other things. So in summary, I think it's important about moving ahead and with all of these different formats, trying to be as strategic as we can with our expression systems, but having a sufficient amount of these systems to meet the needs of a really divergent set of constructs that we're faced with expression. And then, in line with that, increasing the efficiency of our protein production process. And I think they all tie into, both of those ideas tie into one another. But I think I'd like to say that streamlining the harvesting method represents a really good example of that thought process. And with that, I would be remiss not to acknowledge my colleagues here throughout the organization for all of their, for some of their data, for their excellent thoughts and communication. And I'd also like to, very importantly, thank all of you for taking the time out of your busy day to listen to this. And I'm happy to respond to any questions that you might have. Thank you very much.
Thank you, Richard, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window. Type your question into the box that appears on your screen and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, when do you use XP293 versus ExpiCHO for your biotherapeutic development work? And how much is system selection protocol dependent versus protein dependent?
Yeah, that's a great question. So I think in terms of the, early on for us in the sort of early discovery stage, they're sort of intertwined. To answer that question, I don't know. I'd like to think we'll have a better answer to that in about 6 to 12 months in terms of what drives what there. I think that the construct or the protein is going to kind of drive which system we choose. But I'm still not there in terms of making that prediction. But like I said, that is the goal.
Okay, great. The next question we have, thanks for your great talk. Do you think that ExpiCHO expression is somewhat predictive for issues one can suffer later on during cell line development? And could the somewhat better product quality of ExpiHEC be misleading later developability?
Yeah, so that's a great question. I have a good example of that. I think that we had one time where we were expressing a non-antibody that certainly was we were looking at it for as a potential therapeutic. And we expressed it originally in XP293 and expressed very well. And we went through the characterization and decided to move the process forward. When we gave it to the cell line development folks and they put it into CHOs, they came back a number of weeks later and said, look, we can't find anything that is of high enough titer for us to move forward with. And so retrospectively, we went back in and put it into ExpiCHO, and lo and behold, it would have been indicative. I mean, we really got quite low expression, and I think that's it's a great point by the person asking the question. Is that that is a secondary advantage of the system, is that using it as a tool to say that. And we had one other example of that where it was predictive of downstream problems.
Okay, thank you. The next question we have, do you do any labeling of your proteins?
No, I don't. I never have, and so I don't really have any experience with that.
Okay. For the next one, it says, what type of constructs require carrier DNA to be co-transfected?
Well, that's a great question, and that's sort of what we're trying to figure out as well. I think we're putting together, like I said, some bins of different formats that might require that maybe are more prone to aggregation depending on, linkers and what's attached to what. But we're not, like I said, we're not quite there yet with that. I think that right now we're doing that sort of retrospectively where, we can go back and do gene dosing, or with I think the advantage of the small scale stuff, we could even expand that experiment not to only be cell systems, but to do gene dosing experiments at a small scale to really hone in on that as well.
Okay. What is the future of transient protein production?
That's a very good question. So I think it's very interesting in the sense, and this is something, I think is a great discussion to have a whole bunch of people in a room and talk about this. But I think one of the things with the advances in how quickly we're making stable pools, it really puts the pressure on transient protein production to be truly transient. So if we can generate 1 liter expression runs out of stable pools that have gone through selection and scale up within 3 weeks or so, and we compare that to trying to do, say, in the case of ExpiCHO, a 14-day max titer run, we either have a stable pool or we have a single run sort of thing that only differs by 7 days. So I think, like I said, the pressure is on, and this is why we're sort of pushing to move those transient duration of runs to as quickly as possible. So Yeah, I still think there's certainly a future for transient expression, but I think there is going to be continued pressure to continue to optimize the system, whatever system you're using.
Okay. Looks like we have time for one last question. Does the 293 or CHO system express a protein with better native glycosylation patterns?
I'm sorry, Christian, can you say that one more time?
Sure. Does the 293 or CHO system express a protein with better native glycosylation patterns?
Yeah, so we've done some experiments. Actually, we just published a paper about that in Journal of Expression and Purification. But what we find, and we looked at a couple of proteins that we had expressed in production lines versus material generated in 293s and ExpiCHO293s and ExpiCHOs. And certainly the ExpiCHOs are giving us a much better, a more expected glycan pattern than what we see necessarily in the 293s. And some of it can be, I think, a word of caution depending on the protocol that you use. But certainly the data would indicate that we're seeing something that we had hoped for, is that it's more in line with what we're seeing in the production line. And so that was a really good result to see.
Okay. I would like to once again thank you, Richard, for your presentation, and I would like to thank LabRoots and Thermo Fisher Scientific for making today's educational webcast possible. Additional questions will be answered and published on Thermo Fisher Scientific's website. You will receive an email notification when answers are available. Before we go, I would like to let everyone know that today's webcast will be available for on-demand viewing through April of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now, and thank you for joining us. We hope to see you again soon. Goodbye.